Welcome back to this series of videos, Learning from the Core Practicals. In this video, we will carry out an experiment where we extract DNA from a strawberry. But let's start off by looking at what DNA is. DNA is stored in chromosomes. Chromosomes are long coiled up molecules of DNA. There are 46 chromosomes, or 23 pairs of chromosomes, in a typical human cell. And these chromosomes are found within the nucleus of the cell. A gene is a section of DNA on a chromosome. Each gene codes for a particular protein. The order of bases, now the bases you can see on the bottom right hand side of this diagram, where you can see the DNA has been unwound. These bases, the order of them, determines what protein is made. And you should be able to see that there are 4 different types of base here. Some labelled A, some labelled T, some labelled G, some labelled C. And these bases always pair up in a particular way. A always matches with T, G always matches with C. An organism's genome is the word for all of its DNA. So let's do the experiment now. I will talk you through every step and I'll explain what you need to know for every step if you get asked about it in an exam style question. So step 1, what you need to do is we are going to add some buffer solution. In this solution there is salt and there is detergent. Now detergent is basically just washing up liquid. And what we're going to do is we're going to add 10 mils of our solution into a beaker. So I'll measure out carefully 10 mils of our solution and we'll put that into our beaker. Now you need to know the reason for the salt, you also need to know the reason for the detergent. The detergent, the purpose of it is to break down the cell membrane to release the DNA. What the salt does is it makes the DNA stick together. Step 2, what we're going to do is we're going to mush up our strawberry using our mortar and pestle. Okay, so you need to mash it up. So that you've got liquid strawberry. You might have some little bits left, but you really want to mush this up as much as you can so that you have a liquid strawberry. And once that is all mushed up, you can see there, if you come in really close, you can see that's all mixed up and broken down. We're going to add that liquid now into our beaker. And now we've broken down the strawberry and we're going to mix it in with our mixture of detergent and salt. The salt, remember, is what is going to be sticking the DNA together so we can get hold of it, and the detergent is going to be breaking down that cell membrane so we can get access to the DNA. Okay, so now we've mixed that and added it to the beaker. That's step 2 over. Step 3, we've been doing it already, quite naturally actually. You just mix the contents of the beaker well. It's just making sure that we break down those cells so that the DNA can become extracted and we can get hold of it, but we need to break through the cells to be able to do that. So that's step 3, we've mixed it all really well. Step 4, we're going to now filter to remove any insoluble parts, so that would be like pieces of cell. Remember the word insoluble means can't dissolve. So we'll get rid of all the insoluble parts, it'll be left behind in the filter. So I'll show you how to do that. Some people find it a bit tricky to use the filter paper to start with. So I'll show you. If you look closely, we are going to fold the filter paper in half once. we fold the filter paper in half again. Okay, so we've now quartered the filter paper. We definitely do not cut a hole in the filter paper. I've seen people try and do this before, students try and do this. We don't. The whole point is we want the soluble parts to pass through, the liquid to pass through, and we want the solids to be left inside. We only want the liquid. So we place that into our filter funnel like this. You need to hold it down with your finger to start with, just to stop it popping out like that. And we place it into our test tube. And all we're going to do now is take our solution and filter it, put it into the filter. Be careful not to let it come above the filter paper, because then that'll defeat the process. And what we're going to do is we're just going to pause the video now. And as you can see, it started to drip through, but what we'll do for you is it's dripping through quite slowly. We'll fast forward for you and show you what happens afterwards. So we've waited a little while now, and you can see the liquid has passed through the filter paper and the solid strawberry has been left behind, and we've got enough liquid now that we should be able to extract a nice amount of DNA. So I'm going to place this filter funnel aside. Make sure you don't leave it flat on the desk so it doesn't end up making a mess or getting broken. And we're going to move on to step 5, and now we're going to allow the DNA to become visible. Okay, so we're going to use ice cold ethanol to make the DNA visible. Now, this is ethanol. It's a type of alcohol. For it to work, it needs to be kept in ice beforehand. It needs to be really cold. And a point to note here, I often get asked, this is alcohol, is it possible to drink it? Now, first of all, you never drink anything that you get from the science lab. But second of all, this is a type of alcohol, but it is very dangerous. So I'm going to put on my goggles. You can see there are hazard symbols on the bottle. This is not safe to drink, and you would end up having to go to the hospital. It could be fatal. Very likely it would be fatal if you were to drink this. It's really dangerous. And so what you do is you take a very small amount. You'll probably be given a beaker of measured liquid, measured ethanol yourself. I'm just going to pour, and I'm going to tilt the test tube on its side, and I'm going to pour it down the side very gently. You do not want it to go straight into the liquid. And you can see now we have a layer of ethanol and the strawberry underneath. And you should be able to see clearly, as I put in my stirring rod, the layer of white precipitate. Precipitate means solid. Our DNA is that white solid at the top. Can you see I've now extracted some? It's on the very tip of my stirring rod. That is the DNA as a solid that's been extracted from the strawberry. So we've now done the experiment. You've seen how DNA is extracted from a strawberry. Let's have a go at some exam style questions so that you can practice what you've learned from the video today. Where in the cell is the DNA found? This question is worth 1 mark, so you only need to give 1 point. How many chromosomes does a typical human cell have? 1 mark. What is a gene and what does it code for? This question is worth 2 marks, so you need to give 2 points. Why is it useful to use salt when extracting DNA from a fruit cell? This is worth 1 mark. And what does the detergent do to the cell? Please pause the video now, and you can come back, see if you've got the right answers. Welcome back. Hopefully you've had a go at all of these questions. Now let's look at the answers. Question 1, where in the cell is the DNA found? It's in the nucleus. Question 2, how many chromosomes does a typical human cell have? It has 46 or 23 pairs. Question 3. What is a gene and what does it code for? A gene is a section of DNA and it codes for a particular protein. Question 4. Why is it useful to use salt when extracting DNA from a fruit cell? The salt makes the DNA stick together. And question 5. What does the detergent do to the cell? The detergent breaks down the cell membrane. Well done, and thanks for listening to this Learning from Core Practicals video on DNA extraction. Feel free to look at any of the other practicals to revise your knowledge of the core practicals in science.